thanks so much, Francis, for a wonderful introduction. And it is great pleasure for me to be here, thanks to Don Lindberg and Dave Lippman, Jim Ostel, and David Landsman, and the other Genbank people. Here, actually, we heartily congratulate all of you on your 25 years of service, and thank you very much for continuous collaboration with DDBJ as well as EMBL. So this is from all the staff of DDBJ. So from previous speakers, we know that EMBL started in 1980, then Genbank started 1982, then DDBJ, the center was established in 1986. So, of course, the trial input of nucleotide sequence data has started a little bit earlier than that, responding to the request from Europe and the US, but we regard 1986 as a founding year of DDBJ. And there was a tremendous discussion where we should establish DDBJ in Japan, and it was National Institute of Genetics, I think partly because there was Moto Kimura who proposed the neutral theory of molecular evolution. So he was one of my mentors, actually. Here is Mishima, which is Tokyo and Osaka. Then if you have bullet train, Shinkansen, it takes only 1 hour to the west. Then maybe you're welcome to visit DDBJ. Actually, the international collaboration, as everyone is talking, and we had the tremendous privilege as a name because it started from D. So for many years, we had a kind of consensus that it starts from alphabetical order. But to some point of time, the Genbank EMBL started complaining, so let us make it much more random fashion. Now, it's now random fashion. So actually, these are recent years of international collaborators meeting. Like this year, we are prepared to welcome all collaborators to Mishima in the coming May. So in addition to collaborators meeting, also we are going to have advisory board meeting by video conference this year. So as you see, this is kind of a history over the years and nucleotide accumulation. But as you see, the first DDBJ release was in July 1987. The number of entries was only 66. Then the number of nucleotides is 100,000. It occupied about only 0.25%. So right now, DDBJ's share in terms of entries would be like 12 to 15%. In terms of nucleotide, it's 10%. But we are so much grateful to Genbank and EMBL because they treated us as an equal partner from the beginning, even though only 0.25% of share. And this is a kind of a recent activity of annual submission. This one is entries, the red one is the nucleotide. The peak was in 2005, then a little bit reduced, but still, this is a kind of Asian Pacific countries. this is DDBJ, particularly from Japanese contribution. Then, this is Genbank from Asian. This is EMBL. So certainly, we are making certain contribution, particularly from Asia Pacific regions. And we have certain secondary databases, such as genome search. In particular, recently, we installed a new search system, which is called ARSA. You should remove A, sorry. This is really first algorithm. It has been called as Shunsaku. And as you see, thanks to American and the European initiative, a human genome sequencing project was completed. So Japanese also contributed this project, and this is previous Prime Minister Koizumi, the previous minister. So here is Sugawara was attending at the ceremony. Okay, so here is the human genome, and also we really made an important role for a rice genome project. This is only chromosome 1, but also all genome of rice sequencing project. In collaboration with RIKEN, we also conducted so-called phantom functional analysis, annotation of mouse. And actually, when we were asked by Hayashizaki from RIKEN to make a collaboration, we trained 4 annotators at the beginning, then we sent them to RIKEN. It was starting point of phantom. And these are kind of recent publications. Also, we conducted a human full-length cDNA annotation. That was because more than 60% of human full-length cDNA clones has been accumulating in Japan. So with the help of Genbank and the EMBL, particularly David Lippman and Graham Cameron, and we made annotation jamboree. That was really a big one by gathering a total of 120 experts in Tokyo. Then we made annotation of human genes. And this was published in PLOS Biology, the second issue. In particular, one of the subsidiary database from Invitational database was H-Angel. This is anatomical gene expression database, and it contains about 19,000 loci, over 60 different human tissues and cell lines. If you make a greater category of 10 from neural to endocrine, then we can tentatively identify the genes which are specifically expressed in a particular greater tissues when a given clone is expressed at the more than 50% of all categories. So like red for neural and the yellow like blood. So this is kind of a distribution over the human genome. And when you look at the tissue-specific genes, which we discovered as 1,479 genes at this time. In particular, we identified like neural system specific gene to be about 400. So by comparing with genomes has been whose complete genome has been sequenced, we can map each of 389 genes when those genes emerged. So like utilizing those database, amazingly, kind of 1.13 gene per million year has emerged just before vertebrates. So a tremendous gene might have emerged just before vertebrate, particularly when you focus on neural system specific genes. And quite recently, Japanese government started the so-called Target Protein Project, which is Post-Protein 3000 project, and the DDBJ become responsible for making platform of this project. And meanwhile, I would like to point out, as you know, now next generation sequencing machine is tremendous. This slide was kindly given by George Weinstock from Baylor College of Medicine in Houston as one of major sequencing center in this country. So this is 454 machine, Then it almost comparable to 80 conventional ABI machine. So it's tremendous advancement. And as you know, apart from ethics problem, Jim Watson's personal genome was sequenced last year as of May. Also, I think one of the speakers in the symposium, Craig Venter's personal genome was also sequenced like last September. And 1,000 human genome sequencing project has changed to be 2,000 human genome project. I don't know the latest number, but certainly increasing. What I was so much amazed was this press release that was made as of February 11th in this year. So Pacific Bioscience claimed that they are going to sequence human genome in about 4 minutes, only 4 minutes. And this was based on nanobiotechnology. The original paper was published in PNAS in January of this year. So if one single human genome is sequenced completely only by 4 minutes, then Genbank, EMBL, DDBJ really has to think of this kind of technological output. Moreover, what I'd like to emphasize in this occasion is like a paradigm shift is taking place like from the genome revolution to a sequence revolution. The current sequencing technology is utilized, of course, not only genome sequencing, but also gene expression, such as transcription, ESTs, SAGE, and CAGE. CAGE is a very similar technology to SAGE. Only 20 nucleotides from transcription start site can be sequenced by concatenated together. Like mRNA, functional non-coding RNAs, can be also obtained, particularly from sequencing. Also, previously, ChIP-PET, right now, ChIP-seq, by sequencing the binding region of DNA with particular protein, DNA and protein interaction can be elucidated by sequencing. Also, PPI, protein-protein interaction, two hybrid system, whatever yeast or mammalian, And also, in order to know the product, the sequencing is crucial. Then epigenomics, when you like to see methylated site, then you can add the additional nucleotide, then you sequence it, then the complementary nucleotide would be different one from original genome sequence. From here, you can identify epigenetic system. So clearly, I think the usage of sequencing technology has been changing toward the variety of biological phenomena, which are so much important. And taking an example of Japanese project, that is Genome Network project. It started just 4 years ago. Then it will be completed within this year. Again, DDBJ become responsible for constructing Genome Network information platform. When you are interested in a Transregulation Network, this is kind of elementary system, so coding regions, then regulatory system. Then when the transcriptional product is made into a protein, then a protein may have an interaction, then finally it goes to a regulatory system, again coding starts, transcription starts. So this protein might go to the different genes. This is a transcription regulatory network. To understand this system is the goal of Genome Network project. So in particular, those are kind of overview of the present data accumulation. But as you see, cage data, it has been accumulated more than 30 million segments. So this is as of December 2007. So currently, almost 100 million segment has been accumulated. Then those data will be open from DDBJ. Those data are currently open already. So when those 20 nucleotide sequence data, which shows a transcription start site, TSS, this is the TSS distribution over the human genome. Interestingly, RIKEN produced a mouse cage data too. Therefore, even though we have to take into account the chromosomal rearrangement, still we can make a comparison with TSS distribution. And in particular, this is the X chromosome. We conducted a similar analysis to other chromosomes. Then certainly obtained cage data shows highest peak at the left sec TSS. Then it have like a Gaussian distribution. However, as you see, those Gaussian distribution is minus 100 base pair to plus 100 base pairs at the centering left sec transcription start site. So that means over beyond minus 100 and plus 100, then it's very, very scarce. That means we know a lot of transcripts are made over the human genome. In the case of mouse, 78% of non-coding region may be transcribed. But still we speculate that really important transcript may not be so large, even though we know the importance, functional importance of microRNAs, non-coding functional RNAs. But still, I think a lot of transcription is taking place over the region of coding region. So, so this is David Lipman, and this is Yoshio Tateno, who retired quite recently, but still he continued to work with DDBJ. And here, and this is early days of Japanese collaborators and advisory meeting, and those previous director, Dr. Tomizawa, then this is, I think, Graham, so people. And I think this is Jim and Graham. Maybe this is in the EMBL. Those are Mitikogo, one of Japanese advisor, the Graham and the Yoshio. And here, I really thank you so much for continuous collaboration. And really, we owe Genbank and the EMBL to continue the collaboration, particularly from the DDBJ side. In addition to the Genbank at EMBL, I would like to thank Christian Barks and Los Alamos people when they really helped at the early days. So personally, I would like to invite David to this stage. Could you? I really like to give this real one to you, showing our thanks from DDBJ. Thanks so much. Thank you very much. Okay, so really, I would like to end my talk by stating that the DNA should be for world peace. Thank you so much for your attention.